So in this chapter, we're going to look at regulation of gene expression. So an overview of this chapter, we're going to start by looking at control of gene expression. Then we will move on to talk about the cloning of plants and animals. And then finally, we will conclude with the genetic basis of cancer. So let's start off with our regulation of gene expression. So question for you, how do you go from a zygote to a mature individual? Right? If you think about it, we start off as a fertilized egg. We start off as one single cell. So if we look here, here is a picture of a human zygote. This is our one fertilized cell. Now, how do we go from this to this multi-celled organism that has all of these complexities? And so this is something that thoroughly interested me. This is something that I was very interested in, and this is why when I was an undergraduate, I studied developmental biology. I was extremely fascinated in how does that one cell know which end is a head and which end is feet? How does it know left and right? How does it have the cells become different over time? And so throughout this chapter, you're going to start to learn about how we go from a single cell and what then makes those cells become different so that you end up as this multi-celled adult that has different types of cells and different types of tissues. And so below, you're just seeing these fertilized eggs from the frogs, and then the frogs go through changes where they form a tadpole and eventually would then change into a frog. And so hopefully through this chapter, you'll kind of start to understand how cell types become different over time. Because if you think about it, right, all the cells in your body have exactly the same genetic information. All of the cells in your body have the same DNA sequences because all of those cells arose through mitosis. Mitosis is making exact copies of cells. So if all of the cells in your body have the same DNA, what makes two cell types be different? And so that's what we're going to look at. So how do cells become different cell types to form distinct tissues, organs, and systems? The answer is differential gene expression. Different cells are utilizing different genes. So while all cells have the same DNA sequences, only certain genes are turned on or off in a particular cell. And that is what makes cells become different from one another. So while they have the same DNA, they are not producing the same proteins. Remember that the DNA is just the blueprint, but it's actually the proteins that are going to carry out the functions of the cell. And so this comes down to this differential gene expression, regulating which genes are turned on, meaning what proteins are being produced, and which genes are turned off, which means that those proteins are not produced. So when cells become specialized, we refer to that as differentiation. So when we say that a cell differentiates, it means that it becomes specialized in both structure and function. Differentiation is controlled by turning specific sets of genes on or off, meaning, again, which genes are being expressed in those cells. So if we compare a cell in the pancreas to a white blood cell to a nerve cell, so these are 3 cell types. These are the different types of genes. Notice that all 3 are going to have a gene for a glycolysis enzyme because all of these cells are going to need to do respiration to make ATP. So we call these types of genes, you'll hear them often referred to as housekeeping genes, meaning that they're expressed all the time. Those are genes that those proteins are absolutely required, and therefore they're turned on all the time in all the cells. The next gene that we can compare would be the antibody gene. Antibodies are a type of protein produced by your immune system to help fight off foreign invaders. All the cells in your body have those antibody genes, but those antibody proteins are only being produced in the white blood cells. So you would see expression of this turned on in white blood cells, but not in the pancreatic cells and not in the nerve cells. Pancreatic cells, the pancreas, its job is to produce hormones, enzymes for digestion. Nerve cells have a different role. Their job is in nerve conduction, right? Sending chemical signals or sending signals about stimuli that the body is receiving. So antibody genes would be expressed in white blood cells. The insulin gene would be turned on specifically in the pancreas because the pancreas is responsible for producing insulin, which basically serves as the body's signal that glucose is available. So when you eat a meal, for example, when you eat that meal, your blood glucose level will start to rise as your body digests that food. As your blood sugar goes up, your pancreas then, as a result, is going to release insulin. Insulin is going to communicate to the cells throughout the body that, hey, glucose is available. Start transporting glucose into the cells. And so the cells in the body will respond by transporting glucose in and using that glucose to make ATP. And as a result, blood sugar goes back down and we maintain homeostasis. We maintain constant blood sugar. It helps regulate our blood sugar. And so the insulin gene is expressed in pancreatic cells, specifically a type of pancreatic cell called pancreatic beta cells. It's not expressed in white blood cells because their job is not to produce insulin. It's not expressed in nerve cells because, again, their job is not to produce insulin. And one of the reasons that people have type 1 diabetes is because those people who have childhood diabetes, what happens in that case is that they actually have an autoimmune disease. Their own immune system has destroyed those pancreatic beta cells, the cells that normally would express the insulin gene. And as a result, their body doesn't make insulin protein. And so when they eat, their blood sugar stays high because their cells no longer get that signal to transport glucose into the cell. Hemoglobin, that's the last one here. The hemoglobin gene is not expressed in pancreas cells. It's not expressed in white blood cells, and it's not expressed in nerve cells because hemoglobin is a protein that's involved in carrying oxygen in red blood cells. So red blood cells would express the hemoglobin gene, but none of the cell types listed here would express that same gene. So we're going to start to talk about regulation of transcription in prokaryotes. So before we can do that, though, we need to talk about different types of genes. If we call a gene constitutive, a constitutive gene is a gene that is always expressed, meaning that it's always turned on, and that gene is going to produce some sort of essential protein, just like a gene for an enzyme in glycolysis. That is essential. The cells need that. It's always expressed. It's always turned on because the cell is going to require that. Adaptive genes, on the other hand, are genes that are expressed only as needed. It's a way for the cell to conserve energy. And what I mean by that is if we build a protein, if we make a protein, that requires the investment of energy. The cell is not going to want to invest to produce a protein, an enzyme, if it's not needed. And so in that case, those types of genes would be referred to as an adaptive gene. They're only turned on when needed. So for example, if we were talking about bacteria can metabolize lactose. Lactose is a disaccharide. Now, if glucose is available, bacteria will preferentially choose to use glucose. It's a monosaccharide. It's easier for them to digest. However, if glucose is not available, but lactose is, then bacteria can turn on expression of genes that are needed to metabolize the lactose. But they only do that under certain conditions. They only do it when lactose is present and glucose is not. Because if glucose is present, they don't need to make enzymes to metabolize the lactose. So we are going to look at two categories of adaptive genes. We have what are referred to as inducible genes, meaning that those genes by default are off, but they can be induced to be turned on. Or we have repressible genes who default is on, and they have to have some signal that causes the genes to become repressed. So we call these our repressible genes because their default is on, a signal has to be present to then turn those genes off. So let's start by talking about our inducible genes. Again, these are part of our adaptive genes, meaning that they are controlled. They're not on all the time. In an inducible gene, they are influenced by the substrate concentration, meaning the substrate concentration is going to influence if that gene expression is turned on. Now, why do we even have these types of genes? Well, because simply it's used to conserve energy. We don't want to make those enzymes, those proteins, if they're not needed by the cell. Default position of an inducible gene is going to be off. So again, if we call it inducible, it has to be induced to turn on expression. By default, it is going to be off. An example would be the lactose operon, or what you'll often hear called the lac operon. The goal of the lac operon is to produce enzymes to break down lactose only when lactose is present. So again, if lactose is not present, we don't want to make enzymes to metabolize lactose. And so that is what the lac operon is going to do. So in bacteria, we have what are referred to as operons. And these are the transcriptional units in bacteria. They are a cluster of functionally related genes that can be under the coordinated control of a single on-off switch. The on-off switch, that regulatory switch, is a segment of DNA called an operator. And the operator is usually a sequence that's within the promoter. Now, you might recall that when we talked about transcription, that the promoter is where transcription is initiated, meaning that normally to initiate transcription, RNA polymerase is going to bind to the promoter, and that will allow transcription to be turned on. Within the promoter, in bacteria, there are these operator sequences. These are the on-off switch. They will control if gene expression is turned on or off, and you will see this in just a minute. The operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. So again, here we go. You can see our promoter, our operator, and the genes that they control. And so we're going to look at how does this system work. E. coli is a type of bacteria that's normally found in your gut. Everybody has it as part of your gut, as part of your normal flora. Now, E. coli can metabolize lactose in the absence of glucose. So again, if glucose is present, E. coli is going to choose to use glucose as its first choice for food because glucose is a monosaccharide. However, if glucose is absent and lactose is present, E. coli can then metabolize the lactose. However, it requires expressions of genes to do this, and these genes are only expressed when lactose is around. So again, by default, these genes will be off. But if lactose, which is the substrate in this case, if lactose is present, expression of these genes is going to be turned on so that the bacteria can metabolize that lactose. So let's talk about how bacteria can metabolize lactose. Lactose, being a sugar, is polar, right? If you think about if you take sucrose, which is table sugar, and you put that in your coffee, it is going to dissolve because sucrose is polar. Lactose is also polar. So what that means is that it can't cross the cell membrane on its own. It needs the assistance of a channel protein in order to get lactose into the cell. The name of this channel protein is called galactoside permease. So permease means to permeate or to get in. It's what's going to allow lactose to get into the cell. So you have to have expression of that galactoside permease in order to get lactose into the cell. The cell has to produce that channel protein to get lactose in. Lactose metabolism also requires an enzyme called beta-galactosidase. Beta-galactosidase is just basically the fancy name for bacterial lactase. Remember that lactase is the enzyme that breaks down lactose. Whenever you see ACE, you should think enzyme. So the name of lactase in bacteria, it has a specific name. It's called beta-galactosidase. And this enzyme is going to catalyze the hydrolysis of lactose, basically meaning that it's going to break apart the disaccharide. When it breaks apart the disaccharide, it's going to produce galactose plus glucose. And so it's going to break down the lactose, the disaccharide, into its monosaccharides, the galactose and the glucose. Now, when it breaks it down to galactose and glucose, E. coli can then use the glucose for glycolysis, and then they will convert the galactose to glucose, which can be used in glycolysis. And so this is the bacteria's way to be able to metabolize the lactose for food if glucose is not available. But again, notice that in order to do so, it requires that bacteria turn on expression of these genes, the genes that control the galactoside permease and the beta-galactosidase. Those genes have to be turned on. The cell needs to produce those enzymes, and then they will be able to metabolize the lactose. So if we look at the lac operon, again, we have our promoter, our operator, and the genes that they control. In the lac operon, there are 3 genes that are controlled by this promoter. We have our LAC-Z gene. LAC-Z gene encodes that beta-galactosidase. Remember that that's the enzyme that is used to hydrolyze the lactose. LAC-Y encodes that permease. Remember that the permease is the lactose membrane transporter. It's what allows lactose into the cell. And then the LAC-A encodes what's referred to as a transacetylase. This is used to modify toxic galactosides that can result as a product of metabolism of lactose. Now, these genes, these required genes, are transcribed together as one transcript, and they're all under the control of one promoter. So either they are all on, meaning all of those will produce proteins, or they are all off. And it makes sense. Their job is to coordinate in the metabolism of lactose. If lactose is present, we need to turn on expression of all 3. We need to have lactose get into the cell. We need to have lactose be metabolized. So we need all 3. And so they're all controlled by one promoter. So let's look at how this regulation works. So what we have is that we have our LAC-I, and our LAC-I is a regulatory gene. The LAC-I is what's called the lac repressor. So this gene is going to be transcribed and make an mRNA. The mRNA is going to produce a protein, and the LAC-I gene is constitutive. It's always transcribed, it's always turned on. So in the cell, you always have this active repressor. The active repressor can bind to the operator, and when it binds to the operator, it's going to turn off transcription of the lac operon, because now RNA polymerase cannot bind to the promoter and therefore cannot initiate and turn on transcription. And so when this repressor is active, it's going to bind to the operator and it turns off transcription. Now, does it turn it off completely? The answer is no, it doesn't turn it off completely, because this binding of the repressor to the operator is reversible, meaning it binds and then it comes off, and then it binds and it comes off. Sometimes RNA polymerase does get to the promoter and does turn on expression of those genes. And so what you see here is it says basal expression. What that basically means is background levels. It's going to turn on a little bit of expression, and you're going to see in a minute why you actually need a little bit of these enzymes being produced at all times, but it's at a very, very low level. So in this case, we consider it to be off, right? Default is going to be off. That in the absence of lactose, this gene is going to be turned off. Now, why is it important that we have the enzymes expressed at background levels? Well, because we need a way for the cell to receive a signal that lactose is present. So what is that signal? Well, when lactose is hydrolyzed, it's going to sometimes get converted by beta-galactosidase into allolactose. So the cell is going to take that lactose, and sometimes the beta-galactosidase, instead of it breaking it into its monosaccharides, instead it's going to convert lactose into allolactose. This occurs at very low levels. So again, it's not going to occur often, but it does happen. But this is essential for regulation, because what you're going to see in a minute is that allolactose acts as the inducer. It's the signal that says lactose is present, turn on expression. Now, in order to even make allolactose, lactose had to be able to get into the cell. So you have to have some background level, because without it, you would not have this channel protein. And without that channel protein, lactose can't get in to even make allolactose. And so that's why it's important that there is some background level of expression. There is some basal expression, because you need at least a little bit of the permease and a little bit of the beta-galactosidase to be able to even make the allolactose, which is going to act as the lac inducer. So allolactose is the lac operon inducer. And the way that it works is that the allolactose is going to bind to that repressor. When it binds to the repressor, it causes the repressor to change shape. When the repressor changes shape, it now inactivates the repressor. The repressor does not bind to the operator, and now RNA polymerase can. So again, the repressor can no longer bind to the operator. So notice it's blocked, and when it doesn't bind to the operator, now RNA polymerase does, and it turns on transcription. And so in this way, the allolactose acts as the inducer. It is responsible for turning on transcription, and it does so again by inactivating the repressor. And so this only happens, however, when lactose is present, because the only way that you get allolactose is if lactose is present. And so the allolactose serves as the signal to the cell that says, hey, lactose is present. We need to turn on transcription, and we need to make the mRNA for these 3 genes and then make the proteins for these 3 genes. And so again, this is what we call an inducible system. Default position is off. So when lactose is not present, this is going to be off. However, when lactose is present, it's going to form allolactose, which serves as the inducer, and it's going to turn on transcription of these genes. So, question for you. Which of the following does not happen in E. coli when lactose is present and glucose is absent? Red, allolactose binds to the lac repressor and acts as the inducer. Yellow, the lac-I gene is transcribed. Green, the lac repressor binds to the operator. Blue, the lac-C, lac-A, and lac-Y genes are transcribed as a single transcript. Or purple, all of the above occur. So what I want you to do is I want you to pause your video, write down what you think your answer is, and when you're ready, go ahead and push play. We will not go over the answer right now. This will actually be as part of our discussion in our Zoom meetings. So make sure that you have your answers ready to go for these questions. And so this will be the first question. So you can label this question number 1 and then write the color of your answer, and you will discuss with groups what you think your answer is. And so this is your question for lac operon. So summary of the lac operon: the regulator gene codes for a repressor protein. So again, that is your lac-I. The lac-I gene is constitutive. It's always on, which means that the cell is always making the lac repressor protein. The repressor protein binds to the operator and blocks transcription. That is the default position. However, with excess lactose, right, if lactose is present, that will allow the cell to make allolactose. Allolactose will bind to the repressor protein. It changes the binding site on the repressor, meaning it causes the repressor to change shape so that the repressor can no longer bind to the operator, and transcription occurs. And so transcription is going to be turned on. Now, when you went through the last question, hopefully you kind of reason your way through it, right? And so what I mean by that is, if you think about this, right, if lactose is present and glucose is absent, the first thing you want to ask yourself is, in this scenario, if lactose is present and glucose is not, is gene expression going to be turned on or off? That's the first thing you want to ask yourself is, in this situation, if lactose is present and glucose is absent, do we want gene expression turned on or off? And then, which of those is not accomplished in this question? So you want to kind of reason your way through it when you're trying to answer this question. And so when I'm solving this, I would write myself a note. Gene expression would be on, or gene expression would be off, so that I could see which of these would accomplish that goal. So we just talked about inducible genes. Now we are going to talk about repressible genes. So again, in a repressible gene, the default position is going to be on. This can be causing what's called feedback repression. And feedback repression is going to be mediated by repressors. Again, these are proteins that block transcription. And an excess of the end product results in suppression of the genetic apparatus involved in enzyme production. So you might recall back when we talked about feedback inhibition, where a product can feedback and can affect the enzyme at an earlier step in that pathway. And it causes that enzyme to change shape. And when that enzyme is changed shape, it turns off that pathway. This is a little bit different in that we're not just affecting the enzyme itself. We are actually altering the production of that enzyme. Not the activity of the enzyme, but whether or not the cell even produces that enzyme. And so this is a way that we can have a product feedback and turn off production of its own enzyme. The default position of a repressible gene, again, is on. This has slow but long-term effects. And an example that we're going to talk about for this one is that the effect of tryptophan concentrations. Tryptophan is an amino acid. So we're looking at the effect of tryptophan concentrations on genes that synthesize the enzymes for tryptophan production. So notice for the lac operon, that was genes that are used to metabolize or break down lactose. In this case, we're talking about enzymes that are used to make tryptophan. So not break down tryptophan, but instead synthesize tryptophan. So this system is going to be a little bit different because of that difference in one is breaking down a substrate. This one is allowing you to produce that substrate. So let's look at how feedback repression works. So again, in this type of regulation, gene expression by default is on. So just like in the lac operon, the tryptophan operon has a regulatory gene. And this regulatory gene is constitutive. It's always produced, meaning that the cell is constantly making that repressor protein. But in this case, the repressor protein on its own is inactive. And what that means is that it cannot bind to the operator. And when it can't bind to the operator, then RNA polymerase can bind to the promoter and transcription is turned on. So when tryptophan levels are low, so notice low concentrations of tryptophan. So when tryptophan concentrations are low, the repressor is inactive. It can't bind to the operator and transcription is turned on. Now that's going to make mRNA, and then it's going to produce proteins for enzymes that are used to synthesize tryptophan. So when the concentration of tryptophan is low, the cell wants to make enzymes to make tryptophan. Makes sense, right? If you don't have enough tryptophan, the cell wants to make tryptophan. How does it do that? It needs to make the enzymes to make the tryptophan. However, if tryptophan levels are high, well, the tryptophan itself is going to bind to the repressor protein. So notice again, it's normally inactive, but when tryptophan binds to the repressor, it causes the repressor to change shape. And now it binds to the operator. And when it binds to the operator, it turns off transcription. And now gene expression is turned off. So in this case, when tryptophan levels are high, this is when we get our feedback repression. Because if we have enough tryptophan in the cell, we don't need to make enzymes to make more tryptophan. So in this case, the tryptophan itself is going to regulate feedback repression. The product is going to feedback and it's going to activate the repressor. It's going to cause the repressor to now bind to the operator. And now tryptophan production of enzymes needed to synthesize tryptophan are turned off. So if we have enough tryptophan, then we don't need to make enzymes to synthesize more tryptophan. And the result is that gene expression is now turned off. And so this is feedback repression. Again, default in this case is on. And when the tryptophan is present, it is going to turn it off. Because again, we don't need to make more tryptophan if the concentration of tryptophan is high enough. Now again, notice the difference between this and the inducible. In the inducible, we were making enzymes to break down the substrate. In this one, the tryptophan is going to feed back to turn off production to make tryptophan. So notice that in this case, we're not breaking down tryptophan, we're actually synthesizing tryptophan. And so tryptophan will feed back and it will turn off production of enzymes needed to make tryptophan. So summary of tryptophan operon. So with low tryptophan levels, the repressor protein cannot bind to the operator. Remember that the repressor would be inactive and transcription occurs. However, when tryptophan levels are high, tryptophan will bind to the repressor to form a complex that now activates the repressor to bind to the operator. And when the repressor binds to the operator, transcription is now blocked. So again, the product, high levels of tryptophan, will feed back and turn off production of enzymes that are used to make more tryptophan. So that is our feedback repression. So comparing feedback inhibition and feedback repression, they both have where the product is going to feed back to turn off their own pathway. The way that they do that, though, is different. In feedback inhibition, it's going to affect the enzymatic activity, what we refer to as allosteric inhibition. The product actually feeds back and it binds to the enzyme and it causes the enzyme to change shape and it inactivates the enzyme that way. This method is very fast acting, but it's also very short-lived. Feedback repression, on the other hand, is not inactivating the enzyme itself. It's not affecting those enzymes that are already in the cell that are used to make tryptophan. Instead, it's affecting enzymatic production. Basically, it's regulating if the protein is even produced in the first place. This has a slower response time because it's going to take time for this type of regulation, but the effects are going to be more enduring, meaning they're longer lasting because it will take time to turn back on that system. So this is just comparing feedback inhibition and feedback repression. Now notice when you see repression, you should think gene expression. Feedback inhibition is affecting the activity of the enzyme and affecting it that way. So now we're going to move on and look at regulation of transcription in eukaryotes. Now, each cell in a eukaryotic cell will express only a small subset of the genes in its genome, anywhere between about 3 to 5% of its genome. And this is accomplished by regulated gene expression. Control of gene expression can occur at many different stages in the pathway that lies between the transcription of the gene and activity of the protein. The most common and important use strategy for regulating gene expression acts at the level of transcription, meaning which genes are copied by RNA polymerase. And the reason for that is that would be where you would want to start the regulation, because if you regulate it down here, you've already wasted a lot of energy making the mRNA and splicing it, etc. And so the most common way that we regulate gene expression is simply by transcription, which genes are turned on and which ones are turned off. But we will talk about the other ways in addition that gene expression is regulated. So we're going to break this down into 5 main categories in terms of how gene expression is regulated. The first is going to be regulation of chromatin structure. So basically the way that the DNA is packaged within the nucleus. And so that is going to regulate how gene expression is controlled. We also can have regulation at the level of transcription. So notice that there are multiple levels of where this control can happen. And so a big one is going to be at the level of transcription, basically regulating whether gene expression is on or off by dictating whether the mRNA is even produced. We can have what we refer to as post-transcriptional regulation, so processing of RNA, flow of mRNA through the nuclear envelope, etc. These would all be post-transcriptional, meaning the mRNA is made, but it's before the protein is produced, so regulation of the mRNA itself. Regulation of translation, so regulating whether or not that mRNA is going to be translated. Also, we're talking about the breakdown of the mRNA, so how long does the mRNA hang around? And then we have our post-translational regulation, meaning after the protein is made, we can actually regulate the protein after that in terms of how long does that protein hang around? Does that protein need to be modified in certain ways to become active? These are all different ways that gene expression can be regulated. So we're going to start with the regulation of chromatin structure. And so we have briefly discussed one of these methods in an earlier lecture, and that is the formation of bar bodies. So remember that when we talked about sex chromosomes, females, for example, are XX. And so you would expect that if both X chromosomes are being expressed, that in females, they would have a double dose of any of those genes because they have two X's, while males are XY. But that doesn't happen. And the reason for that is because of X inactivation. And that is that early in development, a random X chromosome is going to become inactivated. And when that random X is going to be inactivated, only one of the two X chromosomes are going to be seen. So for example, if we look at tortoiseshell cats are a type of cats that have both black and orange fur. Now, the reason they have this pattern of these orange patches and black patches is because the gene for the fur color is located on the X chromosome. So if we look at the tortoiseshell cats, they have one X that has the orange allele and one X that has the black allele. Now, during early development, one of those two X chromosomes is going to inactivate. And so what happens is that one of those X's is going to pack in very tightly, and it forms that bar body structure. When that chromosome is packed in very tightly, when it's regulated and packed in, it makes it so that RNA polymerase cannot get active to the promoter and doesn't turn on transcription. And so the bar body, the one that has compacted and packed in tightly, that's chromatin structure. The one that packed in tightly is the one that's inactive. So you will end up with the orange allele being active, and you end up with orange patches on the cat. In regions where the orange allele is inactive, meaning that chromosome is the bar body, the black fur is going to be the one that's expressed. So you end up with black patches. And so these cats have this unique pattern of orange patches and black patches. And this pattern is not the same between any two cats because X inactivation is a completely random process. And so you can have two cats that are genetically identical and yet have a different pattern in terms of their fur color simply due to this random X inactivation, because in some regions the orange allele is going to be active and in other regions the black allele is going to be active. So now along those lines, if we think about tortoiseshell cats, can they ever be a male? Are you ever going to see a male tortoiseshell cat? And the answer is no, not if they're just simply XY, which males are. The reason for that is that males only have one X. So either the male is orange or the male is black, but not both, because they only have one X chromosome. So you're only going to see this in the female cats. And so this regulation of chromatin structure, this is going to be a way that we can regulate gene expression. And so the DNA is regulated at the level of the chromatin structure in that the DNA gets marked with what's called a methyl group. And this DNA methylation is going to control the way that the DNA packs in. And so we saw that when we talked about the epigenetic changes, that you can have two twins, for example, and genetically they can have the same DNA sequence, yet have phenotypically differences in that depends on their epigenetic changes, their changes in their DNA structure, which regulates their gene expression. And they could be genetically identical, yet have some phenotypic differences because of this regulation of chromatin structure, these epigenetic changes that basically are going to dictate which genes are turned on or off. So next, we're going to look at regulation of transcription. And so regulation of transcription, eukaryotic cells is a little bit different than prokaryotic cells. And so when we think about eukaryotic cells, remember that eukaryotic chromosomes are going to be linear. They're not circular like they are in prokaryotic cells. Also, the way that they are regulated is a little bit different. Typically in eukaryotes, each gene is controlled by its own promoter. You don't usually see those operons. Remember that in the prokaryotic cells, you saw multiple genes controlled by one promoter. In eukaryotic cells, that's typically not the case. Usually it's one gene, one promoter. Now, the way that it can be regulated is through several means. There are regulatory proteins that bind to various control sequences to either activate transcription or repress it. So if we call a protein a transcription factor, this is a group of proteins that promote RNA polymerase to bind to the promoter. These basically are going to help with transcription. Activator proteins are proteins that bind to DNA enhancers, so not the promoter, but sequences called enhancers, and they interact with transcription factors, and they help form this transcriptional complex. This complex that is going to initiate transcription and helps facilitate RNA polymerase to bind to the promoter. And so you can see here, these yellows, these are the enhancers, and then we have the activator proteins that bind and help bring RNA polymerase to the promoter and stabilize it there. Silencers are going to be repressor proteins that are used to inhibit transcription. They are going to silence or repress transcription. We also have regulation of transcription at the level of control sequences on the DNA. So the promoter, remember, is going to be the site where RNA polymerase binds to initiate and start transcription. RNA polymerase has to bind to the promoter in order to initiate transcription. And so the sequence of the promoter can dictate how this is regulated. We also have those enhancers. Again, those are the yellow ones. Those are sites on the DNA that help stimulate transcription because activators bind it and help facilitate RNA polymerase to bind to the promoter. So now moving on to post-transcriptional regulation, right? Meaning that we're regulating it after the RNA has already been produced. So what we have in terms of our post-transcriptional regulation is we have a phenomenon called alternative splicing. And this can generate different proteins from the same gene. So when you look at the DNA sequence, it's going to have exons and introns. And so the exons are numbered. So in this particular gene, the troponin T gene, there are 5 exons or 5 coding sequences. Exons, remember, are expressed. The light blue is going to be the intron. Remember that introns are the intervening sequences. They are the part that are going to get cut out, and they're not including in the production of the protein, meaning those sequences don't code for the protein. So when the cell does transcription, it's going to transcribe this entire sequence. Exons and introns are all going to be transcribed. So notice that when we look at the primary RNA transcript, it has the exons and the introns. Now, remember that transcription happens in the nucleus, right? We make the mRNA in the nucleus. But where does protein synthesis occur? And the answer is not in the nucleus, but instead in the cytoplasm. And so we have time as the mRNA is going from the nucleus to the cytoplasm to do what is referred to as RNA splicing. And that is to simply cut out the introns. So in RNA splicing, we're going to cut out introns and link the exons back together. Now, notice there are 5 different exons And what can happen is that we can cut that mRNA in a variety of ways that will give us different proteins based on the way that that sequence is going to be cut. And so in the first scenario, we cut out this intron. But instead of going from 3 to 4, it goes from 3 all the way to 5. 4 is completely cut out. So notice that this mRNA has exons 1, 2, 3, and 5. So notice exon 4 is not there. Now let's look at a different option, and let's look at how that could be spliced. And so I color-coded these blue. So we could cut from 1 to 2, so cut out this intron. But now cut from 2 to 4, so cut out exon 3, and then cut from 4 to 5, so that this mRNA is 1, 2, 4, and 5. Notice that because these mRNAs have different exons, that means that they have different sequences. Those mRNAs have different sequences, which means they're going to produce different proteins. And so cells can take a very limited number of genes and splice them in ways so that you can get multiple proteins from one single gene. And so an example is I studied a gene. There's a gene in humans. It's called OVL2. And that particular gene creates 3 different proteins. And we call those different proteins, they're referred to as isoforms. They're different versions of that gene. Interestingly, those different isoforms are expressed at different times in development. So one is formed in embryonic development. One is used in adult skin, etc. And so by using alternative splicing, our cells can take a limited number of genes and cut them in different ways and produce multiple proteins from one single gene. And so that is because of this alternative splicing. Now, can prokaryotic cells do this? Answer is no. Remember that prokaryotic cells, like bacteria, they don't do splicing at all, because in prokaryotic cells, transcription and translation are coupled. There is no spatial separation of those two processes because prokaryotic cells lack a nucleus. So in prokaryotic cells, when they make the mRNA, right away it's going to be translated and it's going to produce that protein. So there is not time to cut out introns in prokaryotic cells. So this process, this alternative splicing, this is unique to eukaryotic cells because, again, we have spatial separation. Transcription happens in the nucleus, so we make the mRNA in the nucleus, and then the mRNA has to come out of the nuclear pore and then gets translated in the cytoplasm. So because of the spatial separation, this process can occur. So this is just an interesting idea to show you something. So we're looking at the estimated number of genes for different organisms. Saccharomyces cerevisiae is the one on the far left. It has an estimated 6,034 genes. That again is baker's or brewer's yeast. Name means sugar fungus that makes beer. So that Saccharomyces cerevisiae has about 6,000 genes. This is a unicellular eukaryotic cell. It's a type of fungus. We can have Drosophila melanogaster, which is a fruit fly, has about 13,000 genes. We have C. elegans, which is a roundworm. It has about 19,000 genes. We have Arabidopsis, which is a mustard plant. That can have about 25,000 genes. These are all types of organisms that are typically used when we study development. They're what we call model systems to study their development. And so, but interestingly, when you look at these, right, notice that the mustard plant has about 25,000 genes. Humans have between 20,000 and 25,000. New genes are still being discovered. So notice that the mustard plant has more genes than humans. And that almost seems counterintuitive because you would think us as humans being more complex, right, that we would have more genes. And the answer is not necessarily, because our cells are really good at doing alternative splicing. We can take a limited number of genes but still produce a wide variety of proteins because of that alternative splicing. A good example of where alternative splicing is used a lot is for the production of antibodies. And so antibodies are produced by this alternative splicing, what we call VDJ rearrangement. It has this group of genes, and they can be cut in different ways so that our cells can take a very limited number of components for the antibody gene, but splice them in a variety of ways so that we could essentially create almost an infinite number of antibodies. And so this is what makes our immune system so amazing, is this ability to do this alternative splicing and to take a very limited number of genes, but still produce a wide variety of proteins, produce a wide variety of antibodies. And so this is also why when we talk about COVID, for example, the coronavirus produces different types of antibodies in different people. It depends on which part of the coronavirus your antibodies are recognizing, and that's going to be different from person to person. And so not all antibodies are the same. You can have multiple different antibodies that can recognize COVID, and it could be different in different people just because of alternative splicing. So, question for you. Again, the answer will not be provided here because we will discuss it in our Zoom session. But an intron is red, a portion of DNA or primary transcript sequence that does not code for the protein. Yellow, the structure in the cytoplasm that is the site of protein synthesis. Green, the coding portion of a length of DNA or a primary transcript of RNA. Blue, a portion of DNA that is not transcribed into the primary transcript of RNA. Or purple, a portion of mRNA that carries instructions for making the protein. And so this will be for question number 2. You're going to write what color you're going to vote, and then you will discuss it with your class meetings during the Zoom session. And so write down your answer, and then in Zoom, we will talk about the answers. So one type of post-transcriptional regulation that we already talked about was alternative splicing. Another post-transcriptional regulation involves the lifespan of the mRNA, meaning how long the mRNA lasts within the cell. The longer it lasts, right, the longer that mRNA hangs around, the more times it can be translated, meaning it makes more protein. So how does the cell regulate the lifespan of the mRNA? Well, single-stranded RNA molecules called microRNAs or miRNAs are used to control the lifespan of the mRNA. Basically, these act to signal and control whether that mRNA is going to hang around or not. So these microRNAs, they are going to be complementary to a particular mRNA. So we have here, this yellow is our microRNA, and it forms a complex with a protein. And that microRNA, remember that RNA is single-stranded, that microRNA is going to bind to a particular mRNA. And the mRNA has a sequence that is complementary to the microRNA. If it is a complete match, right, if all of these bases line up, that is going to signal to the cell to degrade that mRNA. If, however, a majority of the sequence matches, but there's this part that is not complementary, that is not going to degrade the mRNA. Instead, that's going to simply block translation. So it still will turn off gene expression, but it doesn't actually destroy the mRNA. So for our post-translational regulation, we're not going to talk much about regulation of translation, but we're just going to skip ahead to post-translational regulation. So that is referring to once the protein is already made. So there are several ways that a protein can be regulated once it's already produced. We can have cleavage and folding. So this can happen in terms of cleavage, like cutting the protein is what that means. That can happen in the Golgi apparatus. An example of when cleavage is important, the insulin gene, for example. The insulin gene is going to make originally a protein that we call proinsulin. And that form of insulin, that protein that's originally produced, is not active. It has to be cut in very specific ways in order for the insulin protein to become active. So that protein is regulated by when it's cleaved, where it's cut. We can also regulate proteins at the level of folding, because remember that proteins have to be folded into their correct conformation in order for the protein to function. And you might recall back to when we learned about protein synthesis that the chaperone proteins are the ones that are responsible for folding the proteins. And so the protein has to be folded correctly in order for the protein to become active. Another way that proteins are regulated post-translationally would be the addition of these prosthetic groups, these inorganic atoms that have to be added to a protein to make the protein active. For example, hemoglobin. That is the protein in your red blood cells that is going to bind to oxygen, pick up oxygen, and then carry the oxygen through the blood to drop off oxygen at your tissues. The heme group, or the iron-containing portion, that's the inorganic atom. That iron-containing portion is essential. If you don't have that, hemoglobin doesn't function properly. And that's why when patients are anemic, right, when they're not transporting oxygen effectively, one of the things that physicians will do would be to give somebody an iron supplement, because iron is needed in order to make functional hemoglobin. Zinc is needed in what are called zinc finger domains. These are basically these transcription factors, and they require zinc to allow them to bind to DNA. And so it's those transcription factors, those zinc finger proteins, are not active without the zinc. So you have to add these prosthetic groups, these inorganic atoms, in order for the enzyme or the protein to be active. Post-translational regulation can happen in terms of cellular location, so whether it gets to where it needs to go in terms of where that protein might function. And then lastly, the lifespan of the protein. And so we have a structure that's called the proteasome, and the proteasome recognizes tags that are put on proteins that are meant to be degraded. And so what we have is we have this protein here, and it gets tagged with a molecule called ubiquitin. And so ubiquitin gets added to the protein, and that signals for that protein to go into the structure called the proteasome. So the proteasome is going to recognize that ubiquitin tag, and as a result, it's basically the ubiquitin tag is telling the cell to degrade this protein. And so the proteasome is going to break down the protein, and those protein fragments are then released, but the protein is no longer active. And so we can regulate the gene expression by the lifespan of the protein as well. So next we're going to look at signal transduction and how it controls gene expression. So cells can signal to one another to control gene expression. And this is especially important early in development. Cells have to talk. They say, you become this type of cell, I'm going to become this kind of cell. So you need that signaling. Those cells need to communicate in order for development to occur. In order for cells to have differential gene expression, we need to have cells signal to one another. And so what you have here is you have this cell is the signaling cell, and it's going to secrete some signal molecule. So this cell is sending the signal. This cell is going to be the recipient of the signal. So it has a receptor protein on its membrane, and that binds to the signal, and that causes some sort of what we call signal transduction pathway. Basically, it's going to initiate this signaling pathway within the cell. And the signal transduction pathway basically allows for a signal to become amplified so that the response is even greater. And so this is then going to activate this protein, which activates this protein, which activates this transcription factor. This transcription factor, when it's activated, can now go into the nucleus. It can bind to the DNA, and it can turn on gene expression of that gene. So that's going to lead to your response. So you're going to make the mRNA and then make the protein. And so this is cell signaling. cells are going to communicate with one another, and that is going to lead to transcription and turning on different genes. And so again, this is going to be important for development when we talk about forming a multicelled organism. We need to have differential gene expression. That is going to be controlled by cell signaling. Cells have to communicate to say, I'm going to be this, you become that. And that's how the embryo knows what to do with the different cell types. So a series of RNAs and proteins produced in the embryo control the development of an animal from a fertilized egg. So what we have here is, if we look within an ovarian follicle in the Drosophila, they will have this egg and these follicle cells. And the follicle cells are support cells. Now, the egg cell and the follicle cells are going to signal with one another, and that is going to lead to gene expression and growth of the egg cell. It also will lead to localization of head-type mRNAs, meaning it's going to turn on expression of genes that tell the organism that that end is head. And so when this happens, you get this cascade of gene expression. You get fertilization and mitosis, right? So the egg gets fertilized, and that's how the embryo knows which end is going to be the head, which end is going to be the tail, etc. And so there were a lot of interesting studies done about how this process is regulated. And interestingly, normally the head type mRNAs are localized to one end of the embryo, but they found that if they inject it into this side of the embryo as well, they end up with a two-headed fly, because the presence of that mRNA is going to turn on expression of head type genes. And so if you put that mRNA in a part where it's not normally found, it's going to turn on expression of genes that form another head. And so you would end up with a two-headed fly. Now, a homeotic gene is a master control gene that regulates groups of other genes that determine the anatomy of parts of the body, such as which body parts will develop where in the fly. It leads to this, what we call segmentation. And so you get these different body segments, and within these different body segments, we get different genes turned on or off, and those eventually will lead to these different segments in the adult fly. So let's talk about this more. So these homeotic mutations can alter the identity of a segment. So notice that here we have a normal fruit fly, and this is what it would look like. Here we have a mutant fly with extra wings. So it has this duplication event of this homeotic genes. And so you get this alternate segmentation where you now get a second set of wings where legs should be instead. If we were to look at a mutant fly that's called Antennapedia, the Antennapedia mutant has it so that where antenna would normally be, so if we look at the fly head, here would be where the antenna should be. If you take the leg gene and you put it on the chromosome where the antenna gene should be, what you end up with is you end up with leg segments growing where antenna should be. And that's because these are homeotic genes. They regulate certain segments. And if you move that segment gene into a new position, well, now you end up with whatever structures that controls, in this case, the legs, where the antenna should be. And so these are homeotic mutations. So researchers can monitor the expression of specific genes. So scientists can use a variety of techniques to study how genes work together. So we can look at in an embryo, for example, where a particular gene is expressed. What types of cells, what types of tissues would be expressing these particular genes. So what you're looking here, we have what's called nucleic acid hybridization. So what we call in situ hybridization, you'll also hear it called. It allows researchers to identify cells in which a target gene is expressed. And so we use these probes, these mRNA probes that recognize particular mRNAs. and these probes have a fluorescent tag bound to it. So wherever the mRNA is found in the embryo, so here's the embryo, wherever the mRNA is found in the embryo, so this is the target mRNA, here is the probe, and wherever that mRNA is located, you see it fluorescing yellow. So the yellow is looking at this particular gene, and notice it has this very distinct pattern along the length of the fly body. If we look at this one is now using a different probe. It recognizes a different mRNA, and instead of being tagged with a yellow tag, it tags it blue. And so we can do these overlays and look at where these different genes are being expressed in the organism. We're actually looking at the organism to see where a particular gene is being expressed. We can also do another technique called a microarray, and a microarray is used to gather data about which genes are turned on or off in a particular cell. And so what you have is that each of these little dots is a well that contains identical copies of a DNA fragment from a specific gene. And so what happens is that those little wells have those DNA sequences. And again, we use probes that will bind to particular DNA sequences, and we will see which genes are turned on or off. And so these different probes, if we see it light up in different patterns, that is going to tell us which genes are turned on and which ones are turned off, and that tells us something about gene expression. Microarray has been very valuable, especially in cancer research, for example, to look at in different types of cancers, which genes are turned on and which ones are turned off. And this is also really revolutionizing cancer therapies, because before it was kind of this blanket, if you have lung cancer, we're going to try and treat you with this drug. And if you have breast cancer, we're going to try and treat you with this drug. But what scientists are finding more and more is that even within breast cancer, not everybody who has breast cancer has the same types of mutations. Some might have a mutation in gene A. Some might have a mutation in gene B. And by doing these microarrays, we can actually see which basically mRNAs are being altered in terms of their expression. And that could lead to more specific types of therapies, targeting somebody's specific mutations so that you treat with drugs that would be the most effective for that particular person's type of cancer. So now we're going to move on and talk about cloning plants and animals. And so an example of this was cloning in plants. And so what they did was they took a carrot, and they took that carrot and they cut it into very small fragments. And those fragments were then cultured in a nutrient media. Stirring of the tubes caused single cells to shear off into the liquid. So we've released them as to single cells. Now, those single cells that were free in suspension began to divide. And eventually it led to this embryonic plant that was derived from a single cell. This plant could then be cultured on agar media and then later planted into soil. And what they found is that this cell, this one single cell that's found in the adult carrot, this one single cell still had the ability to go back, and it could give rise to all the different cell types in the plant, not just the carrot itself, but the leaves and the flowers and all the other parts. And so what this experiment shows is that differentiated cells, so even the cells that have already been committed to become a particular type, even differentiated cells always maintain all of their DNA, even if they don't express every gene. So in the carrot example, right, the carrot only is turning on a certain subset of genes in the carrot itself, but that cell still had the ability to give rise to all the other cell types, the leaves, the flowers, etc. And so what that tells us is that plant cells remain what we call totipotent. Totipotent means that these have the ability to become any type of plant cell. So again, that plant cell has all of the same DNA no matter which cell types you're looking at, but those cells in the plants are able to give rise to an entirely new organism. And so we say that those cells are totipotent. Now, some animals, such as a salamander, for example, can regrow a part of its body when it's lost, such as a limb. And so a lot of research is going into how is that process controlled? Why can some animals, for example, regenerate structures and others cannot? And so this is a normal animal cloning. This happens naturally. So next we're going to look at what is referred to as reproductive cloning. And so in this technique is called somatic cell nuclear transfer. And so what we do is we take a sheep that basically is going to be the egg donor. And the egg has the nucleus removed. So it's basically just an empty shell with no genetic information. They then took mammary gland cells from a donor, and they took those mammary cells and they fused these mammary cells with this enucleated egg, with this egg that does not have its nucleus. So those two cells were fused, and the fused cells start to develop as an embryo. And now the egg has the nucleus from the mammary cell. It gives rise to an early embryo that is then implanted into the uterus of a surrogate. So notice there's three sheep used in this technique. So the surrogate mother is going to carry that embryo, and it's going to give rise to a lamb, and they called this lamb Dolly. This was the first cloned animal. And the reason they called her Dolly is because if you know anything about Dolly Parton has relatively large breasts or mammary glands. Notice that this lamb that was produced, this lamb was genetically identical to the mammary cell donor. And so what that tells us is, again, this cell is differentiated, right? It has become specialized, and in culture, when you grow it, only gives rise to mammary gland cells. But even that differentiated cell contains all of the genetic information. It still can give rise to an entirely new organism. And so again, this lamb Dolly was a clone of the lamb or the sheep that provided the mammary gland cells. And so this is called somatic cell nuclear transfer. The mammary gland cell is a somatic cell, right? It's a cell that's not used for reproduction. And that was fused with an egg where the nucleus had been removed. And then that could give rise to an entirely new lamb. Now, how effective this technique is, it's not high. It took a lot of attempts before they actually got a lamb to be born from this technique. And that has to do with changes in the DNA as we age. Dolly, for example, Dolly did not live a full life in terms of how long sheep should live. And that's because in these cultured mammary gland cells, they had shortened what we call telomeres, these segments at the end of the chromosome that are repeating. When telomeres shorten, we age. And so because these cells had aged, right, they came from an adult donor. Because they came from an adult donor, the telomeres were already shortened. And so even though Dolly was a lamb, her DNA was already aged at that point. And so Dolly didn't live a very long life because her DNA was from an older animal. However, the older the donor nucleus, again, the lower the percentage of it actually giving rise to developing animals. So they did an experiment where they did that nuclear transplant, but instead they compared what happened if they used cells from a frog embryo versus cells from a frog tadpole. And in the tadpole, they used fully differentiated intestinal cells. So notice that these cells are differentiated. They're older, right? They've undergone more divisions. The frog embryo is a less differentiated cell. And they did the same technique where they took the egg cell and they removed the nucleus from it. And then they took this enucleated egg and fused it either with the frog embryo cell or with the intestinal cell. And what they found is that when they took a cell that was younger, when they did this with the frog embryo cell, most of those would then develop into tadpoles. The technique worked a lot better. However, if you take a differentiated cell, a cell that's already aged in a sense, and you do this technique, most stop developing before the tadpole stage. The technique was a lot less effective because the age of the DNA matters as well. So what we just looked at was reproductive cloning. They took an egg and they removed the nucleus. They then added a somatic cell from an adult donor. They grew that in culture to produce what's called a blastocyst. And a blastocyst is an early embryo where the cells still have the potential to give rise to the entire body of the animal. And so they take this blastocyst and they would implant it into a surrogate mother, and then the clone of the donor is born. That's referred to as reproductive cloning. This can be compared with what is referred to as therapeutic cloning. Therapeutic cloning, the goal is not to produce an entirely new organism, but instead to produce cells that can be used for specific therapeutic purposes. And so the beginning part is the same. You take an egg, you remove the nucleus, you add the somatic cell from the adult donor, you grow it in culture to produce a blastocyst, and then the cells in the blastocyst can give rise to form embryonic stem cells. And then you can grow these embryonic stem cells and you can cause them to differentiate into a certain cell type and then transplant those cells for therapeutic use. So for example, we'll look at an example of using stem cells to produce what are called oligodendrocytes, which are a type of cell that myelinates or produces this protective covering on neurons. And the goal is to use that for spinal cord injuries, to produce those cells to repair injured spinal cells. And so that would be therapeutic cloning. The goal is not to create a new organism, but instead to create cells for a particular use. So an example of reproductive cloning, CC, which stands for carbon copy, is a cloned cat of the cat on the left. So here's the clone, here is the parent. She is a calico cat like her single female parent, meaning she's not the result of sexual reproduction, but instead just a clone of the parent. But the pattern and coloration are different because of random X inactivation. So just like when we looked at the tortoiseshell cats, the same idea happens for the calico cats. They have these unique patterns because of random X inactivation. And so while they are genetically identical cats, physically they look a little bit different because of random X inactivation. So here are some other examples of reproductive clones. So again, we have CC the cat. We have cloned animals that are used for medical use to produce some sort of protein, for example. We have clones of endangered animals to try and keep a particular endangered species going. And so these are just some examples of organisms that are produced by reproductive cloning. And so now we're going to move on and talk about stem cells. Most cells in the adult human body have undergone what is called commitment. And commitment is a developmental process that results in cells whose roles are completely determined. And what that means is that those cells will only give rise to that particular cell type. So most muscle cells have undergone commitment, for example, and hence can be nothing but muscle cells and give rise to nothing but muscle cells. So those cells are committed. They are committed, they are differentiated, they are committed to be a particular type, and they are not going to give rise to any other cell type. Cells in the early embryo that have not undergone commitment can give rise to various kinds of cells. And a relatively small number of cells in the adult body have a similar capacity. And both are referred to as stem cells. And so stem cells have a continuing capacity to produce more cells of their own type, along with at least one type of specialized daughter cell. So again, they can give rise to more of themselves and at least one other specialized type. An example of this would be stem cells in the skin. Those cells have the potential to give rise to skin, to give rise to hair, etc. So those are stem cells. They can give rise to more of themselves as well as other types of cells as well. So this is just looking at different types of stem cells. So we can have adult stem cells in the bone marrow, for example, and the cells that are found in the bone marrow, those can give rise to various types of blood cells. And so these are referred to as adult stem cells because they can give rise to more of themselves as well as multiple lineages of cells. So red blood cells, different types of white blood cells, etc. Embryonic stem cells are cells from an early embryo that, when treated under different conditions, can be induced to differentiate, meaning they can become specialized to produce different cell types. So if you culture them in different cultures, they can be told to become a nerve cell, for example, or a heart muscle cell, or they can be differentiated to become blood cells. So cultured embryonic stem cells have a much greater potential in terms of the different cell types that they can give rise to. So cells from the blastocyst intermast are known as embryonic stem cells. And these embryonic stem cells can give rise to all the different cell types in the adult body. So what you're looking at is that when we have fertilization, a sperm is going to fertilize the egg. We are going to form a diploid zygote. That is one single cell that is diploid. That cell is going to divide into two cells. Those two cells divide to 4 to 8, and when we get to about day 5, we have what's referred to as the blastocyst. And within the blastocyst, there is a group of cells that are called the inner cell mass. The inner cell mass are going to give rise to all the cells of the organism. These outer cells here, these will give rise to extra embryonic tissues, so placenta, amniotic sac, for example, other structures that help support the embryo, but the embryo themselves come from these inner cell mast cells. And these inner cell mast cells can be the source of embryonic stem cells, meaning that those cells still have the potential to become any cell type in the adult body. So why are stem cells so promising? Why do scientists want to study stem cells? Well, because if we talk about cultured, what we call pluripotent means that they can give rise to many cell types. Remember that we talked about that plant cells are totipotent. They can give rise to any cell in the plant. In humans, our cells are considered to be pluripotent. The reason they're not totipotent is that they can't give rise to the extra embryonic tissue, which is what is needed to sustain a baby, for example. So we call our embryonic stem cells, they're pluripotent. They can give rise to multiple types of tissues. So what are the uses for these cells? Well, they could be used to identify drug targets and to test potential therapeutics. They could be used for toxicity testing. A big one is that they could be used for tissues or cells for transplantation. So we could take these cells and we could culture them to give rise to bone marrow, for example. And that could be used for leukemia. So when a patient has a cancer of the blood cells and we need to wipe out that patient's white blood cells, those embryonic stem cells could be used to produce new blood cells, ones that do not have cancer. Stem cells could be used to create nerve cells for Parkinson's or Alzheimer's disease, for example. They could be used for heart muscle cells for heart disease. So if the heart muscle is damaged, they could be used to regenerate new heart muscle cells and then transplant those to fix the damaged part of the heart. They could be used to create pancreatic islet cells for diabetes, because these islet cells, these pancreatic beta cells, are the ones that are responsible for producing insulin. And so in patients who have type 1 diabetes, they are defective. Their pancreatic beta cells have been destroyed, and they don't have those cells that would produce the insulin naturally. And that's why those patients have to have shots of injection or have to have a device implanted that will provide them with insulin. However, if we could take embryonic stem cells and differentiate them and make them into pancreatic islet cells, and then transplant those into patients who are diabetics, then potentially their own body could start to make insulin themselves. Another use for stem cells would be to study cell differentiation. So study what are the chemical signals that tell a cell to become a particular lineage. This would help with understanding prevention and treatment of birth defects, for example. And then we have uses that are not yet to be discovered. There are many other potential uses for these stem cells. So I have a video to show you about the first human embryonic stem cell clinical trial that was approved by the FDA in 2009. This was based on work that was done in a lab at UCI, Hans Kierstad's lab, looking for using stem cells to help with spinal cord injuries. And so I'm going to show you the video and show you the work that they've been doing.
When we first met Emma Clateman, she was 14 years old and had been battling type 1 diabetes half her life.
I have to give myself insulin for every single piece of food I put in my mouth.
3 years later, she's watching the news that she says could bring a cure.
There were times where I thought that I would have to live with this for my whole life. But today, I feel for one of the first times in my life that just may change.
It's a bittersweet moment for Emma's parents who feel 8 years have been wasted.
Science in this country is back, and we are very hopeful.
Also hopeful, California researcher Hans Kierstad.
It's going to result in a floodgate of more researchers, more students entering the field, and greater industry investment.
He's experimented on paralyzed rats, injecting them with embryonic stem cells to generate new nerve cells. The rats walked again. Biotech company Geron has been given FDA approval to study Kierstad's treatment in the first ever human trials to start this summer. Other scientists didn't even try to work within the old federal restrictions, taking their work overseas.
This research has been going on in the United Kingdom, in Singapore, in Japan, China, and of course that other foreign country, California.
Californians voted to spend $3 billion in taxpayer money to fund stem cell research here. And around the country, it was state and private funds that helped to develop hundreds of new embryonic stem cell lines.
Federal officials still have to work out the guidelines for the new research, and they warn any breakthroughs are still years away. Sandra Hughes, CBS News, Los Angeles.
CNN medical correspondent Dr. Sanjay Gupta is also a CBS News contributor. And Sanjay, as we heard Chip say, Congress passed legislation in 1996 that prohibits the creation of embryos simply for the purpose of using their stem cells. If the ban against using tax dollars for this is not lifted, will it hinder progress?
It's a good question, Katie. I don't think it will necessarily hinder progress, mainly because there are several different sources of these stem cell lines. The federally funded stem cell lines, as you've talked about. Also, the private sector has been funding stem cell lines for some time, so they're going to be added to the mix. But I think most importantly, to your point, a lot of fertility clinics have embryos that would otherwise be discarded that might be sources of stem cell lines as well. So if you add those all up, I think you're going to have plenty of embryos for potential stem cell lines.
And as we heard, the only FDA-approved clinical trial for using stem cells involves spinal cord injuries. What other conditions or diseases show the most potential to respond to this kind of therapy?
You know, when you think about these sorts of diseases, you have to think about this idea that these stem cells are going to go in there and fix a discrete problem. So problems like diabetes, for example, where the pancreas is not making enough insulin. Problems like Parkinson's disease, where you're simply not making enough dopamine in the brain. Those are the types of diseases, those discrete diseases that are probably going to be the most responsive. And the same thing with spinal cord injury. My guess is a lot of people talk about Alzheimer's disease, but because it is so global in the brain, it might be less responsive. We'll know more on that in the years to come, certainly, Katie.
All right. Dr. Sanjay Gupta. Sanjay, thank you.
Thank you.
I think the greatest challenge of stem cell research is to take a stem cell that can become anything in your body and make it become one single cell type in large number, high purity. My recent research is really targeting the development of a treatment for spinal muscular atrophy, amyotrophic lateral sclerosis, and chronic spinal cord injury. All three injury and disease types rolled into one because all three are characterized by the loss of motor neurons, those cells that control all the moving parts in your body, from your the movement of your internal organs to your arms, eyes, blinking, mouth, everything. Now, those diseases have a deficit of motor neurons, so we've identified a therapeutic target, motor neurons. The path to the clinic really begins with preclinical efficacy work. Do your stem cells actually effectively treat an animal model of human disease? So we have to pick that animal model, find an animal model or develop an animal model that mirrors in some way the human condition. We can never do that. Never ever has there been an animal model that perfectly reflects the human condition of a disease state. So all of our models are imperfect, which means that we have to use 2 or 3. So once we find the 2 or 3 that are closest to the human condition, then we transplant our cells in there and decide on dosing, on timing, the quantity of volume that we're putting into the disease state. In addition, we've got to determine what we're going to look for. What are the outcome measures? Are we going to look for leg function after spinal cord injury? Or are we going to look for bladder function, bowel function, sexual function, motor control, sensory control, any portion thereof? The point here is that we don't want to miss an effect. So we have to have multiple outcome measures. We often have to design those outcome measures, talk to the field, and pick methods that others can follow and others can replicate so that one laboratory can back up the work of another. After we've got this thing working, we then have to show that it's safe. So again, we have to pick the right animal models to look for safety. Do these cells cause tumors? Do they cause pain? We recently designed a safety study that contains some 160 outcome measures, 160 analyses of a single rat, 60 to 80 rats in each group. That's a lot of time. It's a lot of students. It's a lot of weekends. It's a lot of birthdays. It's a lot of time to determine if these things are safe, if they're working. And now in spinal muscular atrophy, some very smart people have developed a mouse model of that disease, but it dies within 14 days of birth. That's not so bad if you're transplanting mouse motor neurons into a mouse, because everything moves fast in a mouse. They live and die within 2 years. But to transplant our human cells into the mouse, we have human motor neurons that take months to grow from the spinal cord to their targets, yet we only have 2 weeks to test their function in this animal model, a very appropriate animal model for this clinical disease. So you see, every model is flawed. It means that we need a multiplicity of approaches. Sometimes we have to develop those animal models, and then we need other laboratories to be able to replicate our work using those very same models. So there has to be a continuity across labs. Can stem cell technologies reasonably be applied to the human condition in the foreseeable future? Well, it was only 1998 where that human embryonic stem cells were first isolated. In a few short years thereafter, methods of differentiating them became known. Just 2 years ago, we had our first high-purity derivation from human embryonic stem cells done in a clinically viable manner in my laboratory here at UCI. Now we're starting to see the derivation of multiple high-purity human cell populations. Heart cells have been made. Pancreatic islet cells have been made. Oligodendrocytes, motor neurons, retinal tissue. We're starting to see the derivation of useful products and their application to animals. So now what the field is trying to do is figure out how we design the preclinical work. What do we have to do by way of efficacy and safety in order to get these into humans? But we have developed the technologies That's very, very impressive in a few short years. So stem cell technologies will be applied in the very near future to human conditions. It's no doubt.
Why is it that I have a small bruise on my spinal cord and I should live my life confined to a wheelchair? Everyone gets bruises. It didn't make sense to me intuitively that paralysis should be forever. One day we will be successful in turning stem cells into cures. We'll be able to be successful in getting out of this wheelchair. September 10th, 1994 was my day that lived in infamy. I was playing linebacker in football. I went in to make one more tackle and I suffered a C5, C6 implosion of the vertebrae. I went up to grab my friend's hand and I couldn't flex my hand. I went from being able to bench press 430 pounds to zero in a single second.
When a spinal cord gets injured, effectively it's a bruise. Most spinal cord injuries actually are relatively small. At the end of the day, maybe the size of a grape. An injury, even of the size of the grape, has huge consequences in terms of communication between the brain and the spinal cord. The brain can't communicate with muscles. The spinal cord below the injury can't send impulses up to the brain.
The hardest thing is actually not being able to squeeze your wife's hand. It hurts not to be able to pick up your son and to comb your daughter's hair. It's beyond painful. My father and I, Don and Reed, we realized there's a lot of great science going on, but there's science we're scattered. There are people working here, and actually people are kind of reinventing the wheel and even doing some of the same science. And so we actually created the Roman-Reed law. And the Roman-Reed law did have a very great unifying effect for spinal cord injury. Maybe my football career died, but my patient advocacy career was born, and that's, to me, is way more important. To find a cure for someone living with paralysis or to go score a touchdown, I'd rather find a cure any single day of the week. So people living with spinal cord injury should take heart because there's great leaders working right now.
We work at everything from a very molecular level all the way up to working with people with spinal cord injuries. In most injuries, there is some sparing, and that's important. Some of the connections are still there and don't work for whatever reason. And actually, one of the reasons seems to be that they lose their insulation. So the myelin is the insulation, and that turns out to be one of the targets for stem cell therapy that actually is the first one that's coming online, at least in terms of clinical trials.
Oh, the Geron trial, this is so big. This is huge, groundbreaking. We can actually, for the first time, have FDA clearance to be able to take an embryonic stem cell and turn that into a cure.
This actually has just been approved now for of the initial clinical trial, and everyone, of course, is hugely excited about this. And the idea of the Geron therapy that was actually pioneered by Hans Kierstad's work here is to transplant cells that have been differentiated into what are called oligodendrocyte precursor cells. When implanted into the spinal cord, these cells reform the myelin sheath and restore communication. Even a relatively small improvement in function can make a huge difference in quality of life.
Stem cell therapy has helped a paralyzed man regain the use of his arms and hands. I'm going to tell you everything you need to know about this remarkable advancement in medicine right now on I.O. What is up, people? This is Inform Overload, your YouTube source for amazing information, and this information is just that. Allow me to introduce you to Christopher Bozen, the 21-year-old who suffered a traumatic injury to his cervical spine after a car accident. His car spun out of control on a wet road, slammed into a tree and then a telephone pole. When he was in the hospital, the doctors told his parents that he might be permanently paralyzed from the neck down. Spinal cord injury patients undergo a surgery that stabilizes the spine, but it doesn't do much to restore sensory or motor function. Fortunately, doctors also gave him the option of being a subject of a clinical study that might help his condition. A team of doctors at Keck Medical Center of USC announced that they were the first team of doctors in California to inject a patient with an experimental treatment made from stem cells. Led by Charles Liu, the team injected 10 10 million AST OPC1 cells into Christopher's cervical spinal cord last fall. This study is attempting to improve or even restore neurological function. It may not restore function completely, but it would mean that a paralyzed person would be able to regain use of their arms and hands. Doesn't sound like much, but to someone who's paralyzed, it could mean the world. And that is exactly what happened to Christopher. 2 weeks after his surgery, Christopher showed signs of improvement. He was able to feed himself after 3 months, use his cell phone, write his name, operate a motorized wheelchair, and most importantly, hug his family. The surgery has allowed Christopher to be more independent. There is also a very small chance that Christopher might regain use of his legs as well, but doctors don't want to get his hopes up in case it doesn't happen. This surgery is still experimental, but once it's perfected, who knows what's possible. Maybe in a few years being permanently paralyzed will be a thing of the past.
So what you saw in those videos was talking about a therapy that was discovered by Hans Kierstead at UCI, and it was to take embryonic stem cells and have them differentiate into these oligodendrocyte precursors in order to use them for spinal cord injuries. And so in 2009, the first FDA approved clinical trial for human embryonic stem cells was approved. And in 2010, the company Geron bought the rights to do that clinical trial, and they began the first step, which is phase 1, which is basically simply a safety study. It's not actually testing if the therapy is effective. It's simply making sure that the therapy is safe. That's the first phase. Now, during phase 1, they did find that the therapy was safe. However, Geron decided to stop the study due to financial reasons. Other parts of their company was more profitable, and therefore they discontinued that therapy. Others have since picked up similar therapies, and you saw the more recent study, which was done in about 2016, and it did show that injection of those stem cells into a spinal cord injury was able to help restore some motor function in the upper part of the body, so in the hands, and regain the use of arms. So, human embryonic stem cells. So, advantages. Advantages are that embryonic stem cells are totipotent. They're capable of giving rise to all different cell types in the adult body. Now, again, they're not able to give rise to extra embryonic tissues, but they can give rise to all adult cell types. Another advantage is that these cells can divide indefinitely, and therefore one can produce millions of cells from a small population. The disadvantage would be the ethical debate. So embryos must be destroyed in order to make these embryonic stem cells. However, these embryos are from embryos that were going to be discarded by fertility clinics anyways. And so what that means is that when a couple goes in for in vitro fertilization, they take a couple's egg and sperm and they mix them together and they get their embryos. They typically are only going to implant 1 or 2 embryos into the couple that's doing the in vitro fertilization. The other embryos are typically going to be stored in case a couple wants to use them later, or they would be sent to be destroyed as medical waste. And so scientists argue that if you are going to destroy those embryos anyways, you might as well put them to good use and help with human health. And so, the argument that you're destroying embryos, yes, but you're not creating embryos simply to destroy them. You're using cells that were already available, and instead of destroying them as medical waste, repurposing them for research purposes. And then the other debate or the other disadvantage would be that you would have to deal with the body rejecting non-self tissue because those embryos would be from somebody else. And so if those cells are implanted into the body, then the immune system might reject those cells because they are non-self. And so those are advantages and disadvantages of embryonic stem cells. Now, embryonic stem cells stand in contrast to adult stem cells, which are found in small numbers in various types of tissues in the adult body. These adult cells have demonstrated some ability to differentiate into various types of specialized cells, and hence are the subject of continued research interest. However, adult stem cells do not have the differentiation potential of the embryonic stem cells, nor the embryonic stem cells' ability to continue to produce specialized cells generation after generation. Typically, they have a finite number of times that they can divide before they fail to continue to divide. New research in 2007 showed that human embryonic stem cells could be produced without the need to use embryos. And so this should help resolve this ethical controversy. And so what we have is they developed a technique called induced pluripotent stem cells. And so what they did was they took cells, they took fibroblasts, which are a type of dermal cell, and they took human adult fibroblasts, and they basically used a retrovirus to introduce these 4 genes into those cells. And these 4 genes have been known to have association with maintaining stem cells and reprogramming those cells. And so they used a retrovirus to get those genes into those cells. They culture them under embryonic stem cell conditions, and they formed these human-induced pluripotent stem cells, what we call iPS cells. Now, they took those iPS cells and then they studied them to see if they behave as though they are human embryonic stem cells. So they look at things like morphology, so the shape of those cells, expression profile, so which genes are turned on or off, and does that match the embryonic stem cells, epigenetic status, so again, those are the marks on the DNA, in vitro differentiation, so can they give rise to all the different cell types, and then teratomas, which is basically to inject those into mice to see if they form these tumors that basically have multiple lineages in it, which shows that they are in fact pluripotent and they do have the ability to become any type of adult cell. And so I now have a video to show you about iPS cells.
An iPS cell, iPS actually stands for Induced Pluripotent Stem Cell. Complicated name, simple cell. It's a mature cell that has been reprogrammed or back programmed to become what essentially looks like an embryonic stem cell. So the beauty of this is we take a cell that is already developed and already committed to be what it should be, like a skin cell or a blood cell, and convert it back to where it can become any cell in the body. Stem cells have certain genes that are turned on and certain genes that are not. What is done here is genes are introduced into these adult cells, these mature cells. These genes are normally expressed only in the embryonic stem cell. If you put them into this mature cell, they turn that cell into what looks like an embryonic stem cell. The pie-in-the-sky fantasy, which we hope will work, is that a patient who needs a tissue replaced or a cell type replaced could go to the clinic, have, for example, a piece of skin removed, or it might possibly even a swab from inside the mouth where skin cells are available. Have this cell reprogrammed back into what looks like a stem cell, and then have that stem cell now subsequently develop into whatever tissue was problematic. So in this case, maybe a liver or a pancreas or a heart. So you could have a minimally invasive procedure done, slight skin biopsy or taking something from inside the mouth, and replace, in theory, any organ in the body. This is a chance to revolutionize the way patients are treated for many diseases. Right now, the technology is still in its infancy. So there is extensive manipulation of these cells. It will not be cheap to make them. It takes several weeks to back differentiate a cell into a stem cell, and then several other weeks to grow that up so there's enough cells that you can do a transplant scenario. So it takes a bit of time. Depending on the severity of the disease one wants to treat, there could be a drawback to this type of a strategy. If one is having a heart attack and needs immediate care, growing up stem cells over a month or two in culture isn't going to work. However, if the goal is to eventually replace the tissue or replace the organ, such as the heart, and there's enough time allowed, then they would be quite useful. The situation with iPS cells now is that it takes several genes to back differentiate these cells into what looks like stem cells. It's unclear exactly how safe these cells are for the clinical use at this point. Some of these genes have in other systems been associated with malignancy or cancer. So the advantages of the iPS cells are that they are patient-derived and don't require the use of embryos. disadvantages are they do use genes that are known to cause cancer, and it takes quite a bit of time to grow these up. Advantages of embryonic stem cells would be that in theory these embryos are readily available, and if banked appropriately, they could be taken right off the shelf and used almost immediately. So you could cut off the time frame it takes to develop the iPS line. There are advantages and disadvantages of exploring all approaches, and I think it's going to take a few years to sort out which will be the best. And indeed, it may be true that different stem cell approaches may be better to treat different diseases. It is a potentially new way to address many, many different diseases, and I think any inroad we make at any different disease will be spread to others and will leverage so that the field moves forward faster.
So for the adult stem cells, the advantages are that they do not destroy embryos, so that helps get rid of some of the ethical debate. You can make stem cells from the same person that needs the transplant and therefore would not have rejection, because if you can take their own cells and reprogram them to be a different cell type and then put that back into their body, those are self cells and the immune system is not going to reject those cells. The disadvantage of adult stem cells is that it takes a long time to get enough cells to be used for a transplant and therefore would not be useful for, say, a heart attack, which is need to damage or to replace damaged heart cells very quickly, right? You would need to replace that tissue quickly, and therefore waiting to get enough adult stem cells might not be a practical option. And then lastly, there's a high likelihood of developing into cancer because some of the genes used for reprogramming are involved in cancer. And so there have been new types of therapies involved now in iPS cells to try and combat and get rid of some of those genes that are associated with cancer. And my hope is that by basically having you guys find an article about stem cells in the news, that you can discover different types of therapies that they have found by using different types of stem cells. And what we'll see throughout our lifetime is that probably there are different uses for different types of stem cells. Now, question for you. This is question number 3 that you're going to post on Canvas. So question number 3, stem cells are useful because they can produce red, new muscle cells, yellow, committed cells that can replace only one particular type of tissue, green, proteins that are not available from normal cells, blue, new cells that can grow into all types of cells in the body, purple, cells that can be transplanted into other species. So pause your video, think about your answer, and then when you're ready, continue on, and you guys will discuss this question in the Zoom session. So the last part of this lecture is going to be looking at the genetic basis of cancer. And so cancer can result from changes in gene expression. And in our genetics chapter, we talked about different types of genetic changes and how those can lead to cancer. And one type of genetic change that can cause cancer is that we have these genes that are called proto-oncogenes. Proto-oncogenes are basically normal cellular genes that code for proteins that stimulate normal cell growth and division. So these proto-oncogenes will tell the cell to divide. However, those proto-oncogenes are regulated. They are controlled as far as when they are on or when they are off. However, if we get mutations in those proto-oncogenes, that could basically lead to cancer because we can get changes in gene regulation, for example. And so one type of mutation that can occur in a proto-oncogene is that we get a mutation in the gene itself. And when we get a mutation in the gene itself, that is going to change the protein that gets produced, because that sequence is going to tell the cell what protein to produce. So when we get this mutation within the gene, it now takes that proto-oncogene that normally tells the cell to divide but is also regulated, and it takes that proto-oncogene and it turns it into an oncogene. And an oncogene can cause cancer. And so in this case, it might lead to a hyperactive growth stimulating protein. And what that means is that earlier in this chapter, I talked about signal transduction and how cells receive a signal and then that gets transmitted to the cell. So protein A might activate protein B, which activates protein C, and you get this signal transduction cascade. Normally that process is regulated, meaning that the cell is usually only told to divide when it receives the appropriate chemical signal that tells the cell to divide. However, if you get a mutation in one of those proteins that's in that signal transduction pathway, it might affect the protein so that the protein is always active, meaning it doesn't have to receive a stimulus in order for that protein to transmit that signal in the signal transduction pathway. It's always on. It mimics the protein being always on, and it's always telling the cell to divide. That's our hyperactive growth stimulating protein. We could get a duplication event within the genome where this gene gets duplicated and we end up with multiple copies of that gene. And if we end up with multiple copies of that gene, we might get that protein being produced in excess. It's still the normal protein, but now the cell is making more of it. We could also get the gene, the proto-oncogene, under the control of a new promoter. So remember when I talked about types of genes, we have constitutive, they're always on, and we have our adaptive genes, which are regulated. So if we're talking about our proto-oncogenes, those are going to be our adaptive genes. They're only turned on when the cell is told to divide. And then when not, it's going to be turned off. However, if you get that gene, that proto-oncogene, in a new position in the genome, if it gets put in at a different place, and now it's next to a new promoter, and that promoter is for a constitutive gene, meaning one that's always on, now you don't have that control anymore that's turning that gene on or off. That new promoter is always going to be on, and that protein is going to be produced in excess again, meaning the cell is going to continue to make more of that protein than it typically would because of the promoter, which is controlling transcription. Mutations also can happen in tumor suppressor genes. And so a tumor suppressor gene, remember, is like the brakes. That gene is going to produce a protein that is normally a growth inhibiting protein, meaning that that protein is normally there to inhibit cell division. And that helps to regulate cell division so that it divides when it needs to and it turns off when it doesn't. We could get a mutation in this tumor suppressor gene. And that mutation can cause that tumor suppressor protein to be a defective, non-functioning protein. That protein no longer works to inhibit cell division. And now cell division is not under control. We have our unrestrained cell division because we don't have the brakes working. The accelerator is on and the brakes are failing, so the cell is dividing out of control. So when we look at cancer, cancer progresses in a stepwise manner. And what that means is it's not just one mutation that is going to lead to cancer. So if you inherit one particular mutation, it doesn't mean with 100% certainty that you're going to get that type of cancer, because cancer is the accumulation of multiple types of mutations. And as a result, this is why cancer is usually associated with people who are older or elderly, because it takes time to accumulate these mutations. And so if we look at an example of this, so let's say we inherit or we get one change in an oncogene and the oncogene becomes activated. The first thing that might happen at the cellular level is that we get an increase in cell division. So the rate at which the cell divides happens faster. But again, it hasn't progressed yet beyond that. But if we then acquire a mutation in the tumor suppressor gene, so a tumor suppressor gene becomes inactive, that might lead to a growth of a benign tumor. So in this case, we're looking at the colon wall and we're looking at colorectal cancer. And so initially you're going to see this tumor, this polyp, this tumor, that is benign. It has not metastasized. It has not penetrated the tissue to invade other tissue. However, if you acquire a second mutation in a tumor suppressor gene that becomes inactive, that might then allow the cancer to progress, and we now start to see the growth of a malignant tumor. It's going to metastasize, it's going to move and spread to other tissues. And so again, cancer happens in a stepwise manner. It's not a one and done. It's not one mutation and then you have cancer. It's going to be the accumulation of mutations that will eventually lead to cancer. And so again, multiple mutations are needed for cancer. So what you're looking at is that if you look at a normal cell and we get one mutation, it's a slightly abnormal cell, but it's not dividing out of control. We get 2 mutations and then 3 and then 4. And by the time we get to this 4th mutation, we end up with this malignant cell, which causes a lot of problems. Now, one of the reasons that cancer can run in families is that some mutations can be inherited. Now, again, does that mean you're going to get cancer? No. All it does is that instead of starting you here, where there are no mutations, you're already starting one step in. So you already have that mutation, which is going to make it more likely for cancer to occur. And so an example of this would be a gene called BRCA1. And BRCA1 is a normal allele that functions typically as a tumor suppressor gene. So when BRCA1 is functional, its job is to inhibit cell division. However, people can inherit a mutation in their tumor suppressor gene. They get a mutation in their BRCA1. When mutated, there is an 80% risk of developing cancer, usually breast or ovarian, compared to a 10% risk of getting breast cancer for the rest of the population. So notice that by having this mutation, you're 8 times more likely to get breast cancer. So this is why if breast cancer tends to run in your family, so if mom had breast cancer and her sister had breast cancer and grandma had breast cancer, it's likely that they have inherited a mutation in the BRCA1 gene. Now, if they did, right, does that mean you're going to get cancer? Again, no, not necessarily. 20% of people who have that mutation do not. But it definitely increases your risk. And so as a result, some people take preventative measures, and to protect against breast cancer, they might have a double mastectomy where they remove the breast tissue, which then helps to get rid of the risk for getting breast cancer, for example. But again, that doesn't mean you're going to get it. It doesn't mean that you're not going to get it. It just influences your risk. And so like Angelina Jolie, she had breast cancer run in her family, and she took preventative measures to basically try to help ensure that she didn't get breast cancer as well. Now, one of the things that is a little bit challenging about treating cancer is no mutation is found in all cancers. There's not one single type of mutation that we see in every single cancer. There isn't one. If there was, that would make treating cancer much easier, because if we could find one gene that is always mutated, then we could target that one gene to help treat for cancer. But interestingly, cancer is a unique combination of mutations, and even within a specific type. So even if you compare breast cancer, not all breast cancers have the same mutations. They have different types of mutations in different people for breast cancer. So even a breast cancer therapy that might work for one person doesn't necessarily work well for somebody else because they have their own unique set of mutations. So this is just showing you an example. So in this case, we are looking at these 4 genes. We'll call these genes A, B, C, and D. And we're looking at the tumor location. So in the pancreas, for example, it might be found to have mutation B. In the ovary, you might have mutations in A, B, and D. In lung cancer, you might have A, C, and D, etc. And so because of these unique combinations of mutations, again, there's not one therapy that can be used for all cancers. Earlier in this lecture, I talked about microarrays as being a tool to see which genes are turned on or off. And the reason that that can be an advantage in cancer, for example, is that if we do a microarray on a patient's cancer cells and we look at their gene expression, we can compare to see which genes are being abnormally expressed. And depending on which genes are being misregulated, right, meaning that they're not controlled, if that gene is dividing out of control, causing the cell to divide out of control, then what happens is then we can choose a particular drug depending on the types of mutations. And so this can lead to a more personalized approach to cancer treatments, looking for which drugs will be effective against a particular person's type of cancer. Because again, not all breast cancers are the same, not all colon cancers are the same. Each person's cancer is unique and they have their own types of mutations. So this is just looking at the incidence of cancer. So in 2009, the most prevalent type of cancer would be lung cancer. So 219,000 cases in 2009. Estimated deaths, 159,000. So notice that by far, that is the highest cause of death in regards to cancer. In terms of risk factors for lung cancer, cigarette smoke is a big one. That is definitely going to increase your risk of getting lung cancer. Again, does that mean that you're going to? No. Many people have smoked throughout their whole life and not gotten lung cancer, but it definitely does increase your risk. If we look at breast cancer, notice that breast cancer has almost as many cases as lung cancer, right? It has 194,000 cases. However, if we look at the deaths, notice that the death is only about a quarter of the deaths of lung cancer, about 40,600 cases from breast cancer. So notice that if you are diagnosed with breast cancer, the survival rate is much higher. Can you think of why that might be, that breast cancer might have a fairly high survival rate? The answer is it comes down to early detection. For breast cancer, we have several ways which we can detect breast cancer early, because if caught early and treated early, it hasn't spread to other tissues and therefore is much easier to treat. So if you think of breast cancer, the way that that is going to be screened for is that people can do self-exams, right? You can test and feel your breast tissue to see if you can find any abnormal lumps. And then when you become older, the other preventative way that we can detect it would be by mammogram. So using a mammogram to detect changes in the breast. So some other examples. So we have skin, right? And a risk factor for skin cancer would be UV light, so sunlight. Notice that when you look at pancreatic and liver cancer, they have a fairly high mortality rate. So if we look at pancreatic cancer, it has 42,500 cases in 2009. And of those, 35,200 died. If we look at liver cancer, 22,600 cases, estimated deaths, 18,200. Those have fairly low survival rates, meaning that you're much more likely to die if you are diagnosed with those types of cancers. The reason for that is they are difficult to detect. It is difficult to catch those symptoms early on, and typically by the time you have visible symptoms, it is because it has metastasized to other tissues. And so early detection is a really big player in terms of survival rate of certain types of cancer. The better we get at early detection for various types of cancers, the better the survival rate is going to be for that cancer. So cancer is the second leading cause of death after heart disease in most industrialized nations. Most cancers arise from mutations caused by environmental factors. Agents that alter DNA and make cells cancerous are referred to as carcinogens. So these carcinogens are more likely to mutate DNA, and mutations in DNA is what lead to cancer. And so reducing exposure to these carcinogens and making other lifestyle choices can help reduce the risk of cancer. And so what you're looking at with this bottom diagram is that if it's a blue dot, it means that early screening is available. So colon cancer, for example, can be screened with a colonoscopy, right? Breast cancer, a mammogram, or a breast exam, etc. So notice that the other thing, the size of this dot is proportional to its fatality rate. So notice that pancreatic cancer, for example, has a very high fatality rate. And so again, that's because it's not early detection. We don't have a way to detect it early. And by the time that we detect it, oftentimes it has metastasized to other tissues. Lung cancer, right, also does not have early screening, and it is difficult to detect and therefore has a high fatality rate. Breast cancer, right, we talked about breast exams. Cervical cancer, we have a pap smear, etc. And so we have ways to screen for those early, and that helps with the survivability rate. The other thing about this graph is that it also shows you risk factors for different types of cancers. So if I look at breast cancer, for example, the risk factors that are associated with breast cancer would be alcohol consumption, being female, genetics. So again, if you have like that BRCA1 mutation, obesity, cigarette smoke. If we go down and we look at cervical cancer, being female, right, because males don't have a cervix, genetics, viruses. So HPV, human papillomavirus, is a virus that can increase the risk of cervical cancer. And so that's why they have a vaccine. That's why they have Gardasil, to try and vaccinate against HPV to protect women from getting cervical cancer, and that then will protect them. So if you look at this, you'll notice that smoking has a high risk factor for many types of cancer. If you look at this graph, it increases your risk for pancreatic cancer, colorectal, lung, breast, leukemia, what other ones? Oral, cervical, ovarian. Lots of different cancers have their risk increased by smoking cigarettes. And so again, this is just showing you that cancer is a combination of factors. It can be both environmental, it can also be genetic. There are many components that go into whether or not somebody gets cancer. And so this is going to conclude our chapter 11 video on regulation of gene expression.